I just taught with. Okay. Hello, everyone. I'm slightly discombobulated because I just taught in here for 80 minutes. But we'll do this. Okay, so I am super excited to host Rachel Penczykowski, who was my first grad student. So super fun to host her here. We have known each other a very long time. So we met when I was a postdoc at the University of Wisconsin in Madison, and Rachel was an undergrad who was working at the Center for Limnology with Steve Carpenter and a mutual friend, Stephanie Quinn Davidson. And she was also a music performance major, so that's pretty cool. And so we had the great fortune to meet then, and then I had the great fortune that when I started my lab at Georgia Tech, Rachel was my first grad student, so that, like, I won the lottery there. So she did her PhD at Georgia Tech, and I abandoned her for the last part of it to move here. So this is Rachel's second time in Ann Arbor. Her first time was driving all of our clones from Atlanta to Ann Arbor. My first year here, my lab was in the chemistry building in a wing that is now ROTC. So that was Rachel's first visit to Ann Arbor. After she finished her PhD, she did a first postdoc in Finland with Annalisa Laina, and that's where she started working on plant pathogens. I'm very sad she gave up on Daphnia. I'll forgive her. Then she actually did a second postdoc in Madison with Tony Ives, who is my postdoc advisor also, so that's another fun thing we share. And then she moved to WashU, where she was an assistant professor, and she is now an associate professor. So Rachel's amazing. I'm going to let her tell you about her work. She's had, she has tons of cool publications. She's had a lot of support for her research, including multiple NSF grants, including a career grant. So I will let you take it away.
All right, thanks, Med. Is this working? All right, cool. Another fun fact is the very first contact Meg and I ever had, she emailed me about aqua jogging. Which I don't enjoy aqua jogging, but we had something to discuss there. So today I'm going to tell you about work from my lab over the last several years on plant disease dynamics, and especially how those dynamics play out and may continue to play out in a warming world, with insights from our favorite plant pathogen system, which is Plantago host plants and powdery mildew. So as we all know, humans have caused lots of different types of dramatic environmental changes in recent decades, including increases in temperature around the world, and also changes in land use. So while I'm sure the global temperature anomaly graph is very familiar to everyone, which is why I went past it so quickly, I want to focus on this map of the global spatial extent of land use and land cover changes in the 6 decades between 1960 and 2019. So all the areas in yellow are places that have had a single change in land use between any of like 6 major categories of urban, cropland, pasture, forest, etc. The ones that are pink have actually had multiple land use, land cover changes. So as you can see, humans have caused a lot of dynamic changes in what's happening in any given area of land. And so with these and other types of environmental change, there is of course an urgent need to understand the impacts on infectious diseases, including of humans, animals that are not humans, and plants, for many important applied reasons of public health, conservation, and food security. And so my lab studies effects of environmental change on the ecology and evolution of infectious diseases, and also effects of diseases on ecosystems. And so when I say ecosystems, I really mean all of the living and non-living parts of the environment, everything from climate and weather, land use and habitat structure, nutrients, interactions with other species, including host microbiomes. And then the many aspects of disease that we're interested in include pathogen survival and reproduction, transmission, host susceptibility, timing and size of epidemics, host survival and reproduction, host use of resources. So lots of things you can measure at different spatial and temporal scales. And so today I'm going to focus on just that top arrow, the ecosystem to disease direction, and specifically some work that we're doing on climate variation and land use context effects on a fungal foliar pathogen, which is powdery mildew, on these beautiful plants here. So the kind of roadmap of today's talk, I'll give a little introduction to powdery mildews, and then tell you what we've learned about species specificity of powdery mildews in our system. And then I'll tell you about some projects we're working on regarding effects of large-scale climate variation on powdery mildew occurrence and genotypic diversity. We'll kind of focus on a latitudinal transect that we've been studying over the past several years. And then we'll get closer to home, and I'll tell you about a project on effects of urbanization and fine-scale climate variation, like microclimate, on powdery mildew prevalence and severity. So we're going to go on a journey from the global all the way down to the microhabitat scale. Okay, starting with powdery mildews, which luckily some of you already know powdery mildews. They are beautiful fungal pathogens. Powdery mildew is a general term, but there's really about 900 species of powdery mildews. They're all in the family Ericifaceae. They're ascomycetes. They are all obligate biotrophic plant pathogens, so they can only grow on living host tissue. This is a constraint in some sense because we cannot culture them except on leaves. But we make that work. We grow a lot of healthy plants in the greenhouse, and every week or 2 weeks, inoculate those plants with mildew to keep it going. Some powdery mildews have wide host ranges. Others can infect only a few closely related species. But lots of different host plants get infected with powdery mildew. This phylogeny of the powdery mildews is then mapped onto the different host types, and you can see there's trees, shrubs, and herbaceous plants that often get infected by powdery mildews. And powdery mildews are important agriculturally and horticulturally. They cause yield losses, and a lot of money, millions of dollars per year, hundreds of millions of dollars, is spent managing and trying to ameliorate their effects on economically important host plants. Okay, so the powdery mildew life cycle is really interesting. The asexual cycle is what we see during the growing season, and what it looks like is a spore germinates on the surface of the leaf, and these haustoria form and invade the plant epidermal cell. So we're just on the very surface layer of the plant. We're not going any deeper into the tissue. Hyphae then grow on the leaf surface and form sort of a mat or a web of hyphae. And then upward from those hyphae grow these chains of transmission spores called conidia. So these conidiophores look like lovely strings of pearls coming up from the top. And that is what's giving an infected leaf kind of a textured, fluffy appearance. Those spores are then wind dispersed either singly or in little clumps. And they can land elsewhere on the same leaf or on another leaf of the same plant or a plant nearby, or catch a gust of wind and travel much farther to another host plant. Then the sexual cycle can occur if conditions are right. And in particular, this is a strategy for surviving winter or other off seasons. So I mentioned these are obligate biotrophs. They can only grow on living host tissue. If they do not have living host tissue, for example, if all the leaves have died back for winter, this is how they can survive, is that if there are compatible hyphae, they will fuse. I'm going to get back to that compatible hyphae thing in a minute. And they will produce this case called a cosmothecium, inside of which the haploid ascospores mature in sacs within that case. It kind of protects them from the elements. It's darkly pigmented and helps them not desiccate and freeze during winter. And then in the spring, that cosmothecium splits open and the asci are released, and those ascospores can reinitiate the asexual cycle the following growing season. Okay, so let's get back to this point of the, if these hyphae are compatible, they'll fuse and undergo sex. Powdery mildews have a really interesting variety of mating systems. So that relies on mating types. And so there are some powdery mildew species that are called heterothallic fungi, so they are self-incompatible, and each strain contains only a single mating type. Each strain has a single mating type locus with one of two possible idiomorphs, mat11 or mat12. And so for these heterothallic individuals to be compatible and mate, each must have a different mating type at that locus. And so in this case, surviving winter through production of cosmothecia requires co-infection by two strains of opposite mating type. And so this presents an opportunity as well for genetic recombination and novel pathogen genotypes. There are also self-compatible or homothallic powdery mildews, or more generally Ascomycete fungi, but in this case we're talking about powdery mildews, that contain loci with both the mat11 and mat12 mating types in their genomes. And so for them, a single haploid strain can reproduce sexually. And so to give an example of a system where this matters a lot to the possibility for sexual reproduction, I'll tell you a little story about powdery mildew on hops. So hops is, of course, an agriculturally important grain if you like beer. And it turns out that mating type availability constrains sexual reproduction in this system because the powdery mildew of hops is a heterothallic fungus. And so in this part of the US, the Midwest and Northeast, we do see production of cosmothecia in the hops powdery mildew. That's a nice little image of the cosmothecium with all those anchoring appendages that hold it to the hyphal network. But in the Pacific Northwest, where there's also powdery mildew, they do not find cosmothecia. And it turns out in the Pacific Northwest, when folks have sampled a lot of infected leaves, they only see one of the two mating type idiomorphs, and that's the mat11. So they're just missing the other partner needed to undergo sex. Whereas in the place where the cosmothecia are forming, we have kind of equal ratios of the mat11 and mat12. So in this case, it's not so much that the environmental conditions are preventing sexual reproduction from occurring in the Pacific Northwest. It's that we only have one of the two required mating types, and if that other mating type arrived, then the pathogen would have the opportunity for sexual reproduction and genetic recombination through that means. Okay, so this is, of course, again, an agriculturally important pathogen. Most of what we know about plant diseases comes from agriculturally and economically important plants. But our lab studies wild plant pathogen interactions, which kind of counterbalances a little bit the fact that most of what we know about plant diseases centers on crops or comes from studies of like the genetic model plant Arabidopsis under controlled conditions. So yes, very important to study diseases of crops that we need as humans, but it means that we're missing out on a lot of diversity of plant pathogen interactions in diverse environments. And those broader systems could help enhance our basic understanding of plant disease processes. And furthermore, wild plants and their pathogens can be investigated across lots of different environmental variation, because unlike crops whose whether they're cultivated or not in a given place depends a lot on not just climatic factors and soil type, but also human preferences and socioeconomic situations, there are wild plants that can grow kind of continuously regardless of what the humans are doing. And so those are the ones that we study. And in particular, we focus on 3 species in the genus Plantago. These are the plantains, but they are not at all closely related to the banana-like plantain. These are dicots, unlike the bananas. And these 3 species have pretty broad geographic ranges. So Plantago lanceolata and Plantago major are native to Europe and Asia, and traveled the world with European colonizers and now have a cosmopolitan distribution. Whereas Plantago rugelii, which looks a lot like Plantago major, they can be tricky to identify, but not at all impossible to tell apart. They are native, endemic to eastern North America, but very abundant here. And so we study all 3 of these in part because we very frequently find that they are infected with powdery mildew. And it turns out that there are 2 species of powdery mildew that are known to infect plants in the genus Plantago. That's Golovinomyces sordidus and Potosphaera plantagenus. Potosphaera plantagenus has been extensively and intensively studied on Plantago lanceolata in Finland. That's where I did my postdoc. We've learned a lot about that system from the work there. And it is an example of a homophthalic powdery mildew that's capable of self-mating. Golovinomyces sordidus has been less well studied to date. And as I'll show you a little bit later, we have found that it's heterophthalic. So it requires co-infection by the 2 different mating types in order to undergo sexual reproduction. Okay, so I'll tell you just quickly what we know so far about the species specificity of those 2 mildew species, and then we'll get to some of the climate variation work. So in asking the question, is there any species specificity here, our first clue came from just good old-fashioned observational field surveys. And so we started to go on these road trips, finding populations of Plantago and looking for powdery mildew. And so what I'm showing here in these gray dots are places where these different pairs of Plantago species co-occurred in the same little area of a park or whatever, and where we really exhaustively searched for powdery mildew on both species. So all 3 can be found kind of along this whole latitudinal gradient, but we see them kind of in different pairings here and there. And so now I'm going to color code the points by whether or not we found powdery mildew at the same time at the same place on those pairs. And so if it's a black dot, that means both species were infected on that survey. If it's a white dot, neither were infected. And if it's a color, that means one but not the other was infected. And so what we have found is that where Plantago major and Rugelii co-occur, there's a lot of black and white dots there and relatively few pink or orange on the leftmost panel. So it tends to be like they're both infected or nobody's infected. But where Plantago major or Rugelii co-occur with lanceolata, very often Plantago major or Plantago Rugelii is very infected. I mean, like 90+% of plants have powdery mildew, and the Plantago lanceolata, very abundant, literally physically touching those other Plantago congeners, are completely uninfected. And this is a quite significant effect and discrepancy of infection for the lanceolata pairings with major and Rugelii. So this was our first clue that maybe lanceolata is not getting infected by the same things that infect major and Rugelii. So then we developed some species-specific primers to figure out what species of mildew are we even seeing? And it turns out that Golovinomyces sordidus infects all 3 species. That's the kind of teal color there. But we've only ever detected Potosphaera plantagenus on Plantago lanceolata. Okay, so that's definitely another hint that there's some specificity here for Potosphaera plantagenus. But we still, at the species level, see Golovinomyces sordidus on all 3. So is it the same strains of Golovinomyces sordidus on all 3? Or is there some within species specificity? So to answer that question, we collected seeds and mildew strains from all 3 species across several populations and did a big inoculation assay. So the results of this inoculation assay are going to be shown as the Golovinomyces sordidus strains on the x-axis, isolated from either lanceolata, major, or Rugelii, inoculated onto plants of those 3 species shown on the y-axis. And it's going to be a matrix of squares. Red means infection. So darker red, more heavily infected result. Okay, so what did we find? We found that the mildew strains isolated from Plantago major and Rugelii grow really well on their own species and the other species. So consistent with our field observations of these 2 tend to be infected in the same place at the same time, they can literally share the same strains with each other. When we isolate Golovinomyces strains from Plantago lanceolata, it tends to grow quite well on other lanceolata lines. Not really very well on Plantago major or Rugelii, sometimes a little growth, but except for a few very unlucky Plantago major genotypes, they're able to at least limit the amount that grows. However, to date, we have never, ever been able to get Gallinomyces sordidus strains from Plantago major or Rugelii to infect Plantago lanceolata, even though we have done this experiment and like, it's just like a constant quest. I'm like, sometime I want to make it happen, but we've never been able to transfer mildew from major or Rugelii onto lanceolata. So that's this cool specificity with a little bit of like interesting asymmetry that we don't yet understand, but will be an interesting thing for us to figure out at some point. We have also done due diligence and done lots of experiments trying to get Potosphaera plantagenus to grow on either Plantago major or Rugelii, and this slide is outdated, but in all of our testing that we've done, we've never gotten it to grow. So not only do we not detect Potosphaera on major or Rugelii, when we paint it on in the lab, nothing happens. So this is our like working model of the specificity in this system, is that Potosphaera plantagenus only infects Plantago lanceolata. Gallinomyces sordidus can infect all three, but it's certain strains on major and Rugelii and certain other strains on lanceolata. So is this, yeah, we're staying out of the fray of like, should we call it something different? We're just saying it's different strains for now. Okay, so moving on, what have we been looking at with respect to large-scale climate variation in this system? So one of our NSF grants is studying large-scale climate variation impacts on mildew occurrence and genotypic diversity, and the questions that we're asking are, how does climate shape the geographic distribution and seasonality of mildew on Plantago around the world? Are powdery mildew strains locally adapted to temperature or adapted to host genotypes along a focal latitudinal gradient? In what geographic regions and seasons can powdery mildew undergo sexual reproduction? And then how does climate affect resulting powdery mildew genotypic diversity? The last one I can't answer yet because we're very much working hard on sequencing the genome of our mildew, but I'll give you a sense for the first three questions of where we're at. So one way we can ask questions about powdery mildew around the world leverages iNaturalist. So it turns out lots and lots of people around the world have snapped photos of Plantago and uploaded them to iNaturalist, and often they're infected with mildew, and it's very reliably, we can reliably visually diagnose powdery mildew, which is a major advantage of this type of plant pathogen. And so we're combining our own field surveys along our focal latitudinal gradient with this citizen science data provided by iNaturalist. And one thing we've done is partnered with a collaborator at Truman State University who built some course-based undergraduate research experience modules where he's had students, lots and lots of students, screen images for powdery mildew and herbivory and a few other interesting things. And they've gotten through tens of thousands of photos of Plantago. And so far, we've got over 2,500 geotagged photos of powdery mildew infected leaves that really are all around the world. Yes, they are biased towards North America and Europe, but we have at least some occurrence records on all continents except, well, Antarctica. I took those photos yesterday in the Arboretum, so I will be adding them to the iNaturalist database. So it's one of those nice plant pathogens that you can find in lots of places. We're then working with my collaborator Adam Smith at the Missouri Botanical Garden and our former postdoc Cedric Ndinga Munyanya. They're doing some species distribution modeling of the powdery mildew of Plantago using this database, and of course accounting for the underlying distribution of the host plants and geographical biases and who uses iNaturalist in the first place. So that work is ongoing. We're really excited about it and really pleased by how many people have photographed Plantago for iNaturalist. And then in our focal latitudinal transect, we've done a bit of work looking for the signature of local adaptation of the pathogen to either temperature or host genotypes. So this was a project led by my former PhD student, Quinn Fox. And so what she did was she sampled seeds and powdery mildew strains from 5 locations that span, a beautiful spectrum of climatic profiles, with, central to northern Wisconsin being quite cold in the winter and nice in the summer, and then like southern Mississippi going from warm to very hot. And so in this project, what she did was tested all the pairwise combinations of host genotypes and pathogen strains. So this was done on detached leaves on Petri dishes. So each plate had a single pathogen strain denoted by the X, and then all the 1 sympatric and 4 allopatric combinations times 3 replicates for each of the 5 strains, and then placed at 7 different temperature treatments in growth chambers. And then she did a lot of sitting at the microscope looking for signs of infection to develop. And so I'm going to show you a couple cartoons of what we might expect to see in our results if the pathogen was locally adapted to either hosts or to temperature. So this was our prediction if the pathogen strains were locally adapted to the host, is that this sort of diagonal line of the pathogen strain latitudinal rank on the x-axis and the host plant line latitudinal rank of the y-axis. The diagonal line would be, bright red with high infection levels, and that the combinations from more geographically distant locations would show less disease, and that this would, perhaps be across all of the temperatures. However, if the pathogen was locally adapted to temperature, we expected to see something more like this, where at the very high temperatures, maybe it's too hot for the powdery mildew to grow, or maybe just the most southern strain can grow, and that maybe the more northern strains grow better at a bit cooler temperatures. And in fact, our results are pretty consistent with this slide. Of course, it's real data, so it's not quite as clean. But we did a bunch of model fitting for models with and without different interactions of the host effect and the temperature, et cetera. And the best model includes like a main effect of host line, but there's no host line by pathogen strain interaction. So no evidence of local adaptation or maladaptation to host lines. But to better visualize this temperature adaptation, I'm going to show the data a different way, and that is through thermal performance curves. So since we had each of the combinations replicated at 7 temperatures, what we did here was we looked at what was the maximum amount of growth for each pathogen strain at each of the 7 temperatures across all of the host lines it was tested on. And so we generated these nice, pretty thermal performance curves, and then we estimated from those the thermal optimum for pathogen growth, which is the dashed line. And so we then took those dashed line intercepts with the x-axis and plotted them against latitude, and we see this really nice pattern of the strains from farther north having cooler thermal optima than the strains from further south. The panel on the right is just the slopes of our bootstrapped linear regressions to show that we, did our best to really find the best slope, which is a negative minus 0.26 degrees Celsius per unit increased degree latitude. And so this was a really interesting result. And what we're concluding from this is that yes, the pathogen strains were locally adapted to temperature. And those estimated thermal optima really spanned quite a decent number of degrees Celsius, ranging from less than 17 degrees Celsius to 20.6 degrees Celsius. So that's a meaningful amount of difference in their thermal optima. There was no evidence of either local adaptation nor maladaptation to sympatric hosts. But when we think about the implications of this, since powdery mildew spores can disperse long distances via wind, I think this suggests that northward spread of warm-adapted strains may allow the powdery mildew to track climate change as the northern sites get warmer. So I would say in this and similar systems, I think it shows that powdery mildews really have capacity to keep up with climate change. And unfortunately, it's not the case that with warming, we're gonna lose powdery mildews in certain regions because it's gonna be too warm for them. Yes, it's too warm for the strains that are currently there, but I think that the warm adapted strains will make their way. Okay, so like I mentioned, we're not interested only in the asexual cycle, but also in the sexual life cycle. And in particular, we want to know in what geographic regions and seasons can sexual reproduction even occur, especially for Golovinomyces sordidus, which is the species that requires co-infection of the two mating types. And so one thing we've done is, again, collected pathogen strains from lots of places and put them together in inoculation assays. And this was a study we did a few years ago where we tested 9 single strain and all pairwise co-infections before knowing which mating type, which strain had which mating type. And first of all, we found that at the warmer 2 temperatures, 20 and 23 degrees Celsius, we didn't see any production of the sexual cosmothecia. We did see both kind of immature in yellow and mature in orange cosmothecia forming at the cooler temperatures, but only for the combinations of strains which we later determined to be through PCR to be opposite mating types. And so this really was a situation where none of the same mating type combinations produced cosmothecia ever. We've then done a lot more screening of specimens collected, again, from our focal latitudinal gradient, and have found that both mating types are present kind of throughout the range in variable ratios, but it doesn't seem that this is similar to the hops system where only one mating type is present in a particular region, at least in this transect. They're both present, and that probably isn't a big constraint on whether or not they go through sex. Now, we are currently running, there's actually a fabulous undergrad named Elizabeth Grieve who is running a big experiment right now, testing for thermal adaptation of the production of cosmothecia in a big growth chamber experiment. So she's got all these different pairwise inoculations put at different temperatures, and we're hoping to find out from that experiment if the northern strains have for example, a cooler thermal optimum for producing cosmothecia than the ones from down south. So very excited to see those data when they're available. Okay, so we're next gonna get to a bit more local scale with our urbanization project in the St. Louis region. And in this project, we're studying how urbanization and very fine scale climate variation affect mildew prevalence and severity. And specifically, part of this involves quantifying climate variation itself across our urbanization gradient, and especially looking at temperature variation in vegetated habitats, and I'll explain in a minute why that's interesting. We're also asking how powdery mildew dynamics vary within and between a whole bunch of populations across a gradient of urbanization, trying through experiments to resolve why are there larger epidemics in, spoiler alert, in more urban populations, and then ultimately wanting to know how pathogen genotypic diversity and population genetic structure are affected by urbanization. Again, this last question we're not ready with yet, but I'll tell you where we're at with the first 3. So this is a map of the greater St. Louis region. We've got the Mississippi River squiggling along on the right. Illinois is to the right of that blue river. The gray is urbanization, with darker gray being a higher intensity of developed land. Forest is green, yellows are pasture and crops. And so we really do have a nice gradient of urbanization intensity going from the rural outskirts through suburbia into the city of St. Louis, which is kind of in the upper right of the graph, or of the map, sorry. And in St. Louis, we would expect, as in most other big cities, that there would be this phenomenon called the urban heat island effect, where the cities are in a city, you're both producing and retaining more heat due to human activities and impervious surfaces which trap heat and then release it, especially at night. And so, yes, we do have an urban heat island effect in St. Louis. This is an land surface temperature map around noon on a single day in September 2022. And so you can see that, bright red city of St. Louis and slightly cooler temperatures in the suburbs and substantially cooler in the rural outskirts. So this, my lab was not involved in the generation of this land surface map, but we are involved in a project with the collaborator Fengpeng Sun at University of Missouri, Kansas City, and my colleague Kim Medley at the Tyson Research Center at WashU, doing a combination of things, including dynamical downscaling of climate models in the St. Louis region. So this is a simulation of a particular heat wave event that we had a couple years ago, where the predictions are for there to have been much warmer temperatures right in the city of St. Louis, and it's been validated with what the actual temperatures were. So we have an urban heat island effect, but again, the temperatures that we're validating with, or the temperatures that we're measuring from satellite-based measurements, are really capturing, not necessarily capturing the habitats that a lot of organisms are using. A lot of, most weather stations are at airports or at other places with a lot of impervious surface, and surface temperature isn't necessarily telling you what's happening near the actual ground in a vegetated space. And so, but we know, all of us, from experience, lived experience, that there are huge swings in temperature as you walk through, for example, a park, cutting through this sidewalk in the shade or the sun, getting onto the grass, being closer to this body of water or farther. And so we have, in this part of this same climate project, we've been measuring temperature in vegetated habitats in a few different ways. Our most low-tech way involved me going out during 2020 to a lot of colleagues' yards with the cheapest possible data logger, burying them just below the surface, retrieving many of them, but not all of them, and then doing what we could with the data, which we recently got published. And we did detect, even in this sort of low-cost way, an urban heat island effect. This was a year-long measurement period, but we detected an urban heat island effect in the summer months, and stronger in the shade, actually, which was not my prediction, and stronger at night. So that's consistent with what people find in cities all over the world, that this urban heat is more pronounced when those impervious surfaces are radiating heat back. We have part two of this project involved much fancier, more expensive sensors, put in much more deliberate microhabitat types of single tree, stand of trees, prairie, lawn, and I'm not going to spend time showing you those data now, but happy to talk about them later with anyone who's interested. Because the whole reason that, well, there's many reasons we're doing this climate study. I think it's interesting, and it's a fun way to collaborate with folks studying urban heat in all sorts of different contexts, human-centric and animal and plant-centric reasons. But we want to know how it relates to powdery mildew, too. And so along this same urbanization gradient, my lab has been studying powdery mildew dynamics on Plantago in 22 parks that span some nice variation in the percent tree cover, the percent impervious surface, artificial light at night, temperature, et cetera. There's some meaningful environmental differences between them, just at the site level. And so we go out every month during the growing season, and we've been doing this since 2019, and we collect data from 50 random plants and their immediate surroundings, data on powdery mildew, on herbivory, flowering phenology, a few other things, We also then collect seeds and mildew strains for lab studies, and for we collect infected leaves that we also will eventually use for genotyping, but in the meantime, we can tell if there's any of those sexual structures on them or not. So lots of data from these studies, and we have a good time. Sometimes it's very hot, but other than that, we have a good time. And so the first hypotheses we tested from kind of our first complete year of data in 2020, we tested the hypothesis that there would be more powdery mildew in more urban sites, for example, due to greater pathogen dispersal between those more highly connected sites. So these plants grow really well along roadsides and in human disturbed habitats, so we expected that they and their pathogens may have greater connectivity more in the city. And we also hypothesized that there'd be more mildew in the shade due to just more moderate temperatures and less UV, which can damage fungi. And we did this for both Plantago lanceolata and Plantago rugelii. So these are the monthly infection prevalence data from our rural parks in yellow, suburban in green, urban in purple. I'm going to use the same color scheme for the rest of the talk. Plantago lanceolata is the top row, rugelii is the bottom row. And infection prevalence is almost always much higher on rugelii than lanceolata. Oh, we don't have much Plantago major in St. Louis for some reason. So these are the two plants we focus on locally. But yeah, by the end of the epidemic, infection prevalence was almost 5 times higher on Plantago rugelii than lanceolata. And then to test our urbanization and shade hypotheses, we of course fit a whole bunch of statistical models, and I'm going to give you the highlights from those. So our urbanization hypothesis was supported for Plantago rugelii. We saw larger epidemics in the more urban sites. So suburban sites had bigger epidemics than rural sites, and urban sites had bigger epidemics than rural sites. This was not a significant trend for lanceolata, which also just had less disease overall. And our shade hypothesis was supported for both species. So quite a bit more disease in the shade than the sun within sites. We also, there's lots of things that may be different about where a plant is in the shade versus the sun in a particular park area. For example, the lawnmowers might try to avoid hitting a tree, so might kind of skirt around the outside of a tree. A tree may kind of physically trap spores in a different way compared to an open area. So we also did an experiment using a solar panel array as a shade treatment in an otherwise big open field. We put plants there, we inoculated some of them, let others get infected if they're going to be infected. And this was one of those experiments where the results were so clear that statistical models kept having a really hard time because there was complete separation, because we didn't find any disease in the sun. Lots of disease in the shade in this experiment. And so this was really confirming that there is something to the shade itself which promotes powdery mildew. It's not just tree presence or fenced presence. So these patterns have been super consistent for the years since 2020. These are data that my current postdoc, Kaylee Arnold, is working on analyzing for lanceolata, again, on the top, rugelii on the bottom. Here we're summarizing the infections as area under the disease prevalence curve, so the integrated infection prevalence from 2020 on the left to 2024 on the right. 2025 data are still being collected. But we see these really consistent patterns of a lot more disease on rugelii than lanceolata. In fact, I want to point out that the y-axis is scaled differently here. So that's 0.5 at the top of the lanceolata y-axis. On the rugelii goes all the way up to 1. Also consistently more infection in the shade than the sun, and also a consistent pattern of more disease in the suburban and urban sites compared to rural. But you'll also notice that there is some variation between years in the total amount of disease, and so that's what Kaylee's really digging into right now. I will say unofficially that it seems that temperature does play a really strong the temperature variation between years in the summer months seems to play an important role in predicting how much disease, and in the negative direction, so hotter, less disease, which is consistent with shade being super important. Okay. So to conclude this kind of observational studies from St. Louis, consistently larger epidemics in more urban sites, strong positive effect of shade on mildew. It's kind of an interesting paradox, though, because plants are more likely to be infected in the shaded microhabitats, but there's less shade overall in the city compared to the rural, more forested area. So it really highlights the importance of kind of the scale of inquiry, because we still have more disease in the more urban sites overall. And so there's, a lot to still figure out here of the why for these patterns. And, one question we have to ask is, are the urban plants just more susceptible to infection? Is that why we see more disease in the city? Or are they made more susceptible owing to differences in soil chemistry, air pollution, frequency of disturbances like mowing? Or is it that since the urban sites are better connected by roads, they have more pathogen spores arriving to them by the disease sort of spreading along the roadsides, or physical movement of spores with air currents on roads, which has been found to be a physical phenomenon. And so to start to untangle these different possibilities, we of course have to do experiments. And the first big experiment we did was led by my graduate student Cheyenne Morris, asking what are the direct and indirect effects of urbanization and microclimate, specifically shade, on disease severity after controlling for a bunch of things that we can control. So I'm just going to preview a hypothesized structural equation model, and we're going to return and dig into it. But there's a lot going on here, which is why I wanted to just prepare you in advance emotionally. Okay, but what she really did was she collected, this is just Plantago regalia in this experiment. She collected seeds across the urbanization gradient. Those are the white circles in the middle of other shapes. And then she grew them and replicated common garden experiments, which are the squares. So there's 6 squares, 2 rural, 2 suburban, 2 urban. And we allowed powdery mildew to just naturally arrive if it was going to arrive. All of these gardens are the same size. They have the same store-bought soil put in. The plants were all started in the greenhouse, transplanted, within a day or so of each other. And each garden has a shade side and a sun side, and then a temperature logger a nice temperature logger in the middle of each half. So we're standardizing soil, host density, and we've got replicate urban, suburban, and rural plants on each half of the garden. And then it's 2 rural, 2 suburban, 2 urban. The suburban and urban gardens are super fun because they are our jumping off point for some community engagement, especially with K-12 schools. So our suburban sites are at school gardens. And so we've developed some curriculum materials that we use with classes, and which teachers now are using on their own to engage students in an authentic research experience. We have collected data even with kindergarteners. Not always easy to see what number is what, but they did measure the plants. Happy to talk about this more later if anyone's interested. But let's get back to what have we found so far as I wrap up here. So again, this was kind of our hypothesized effects that, the sites with a greater % urban area surrounding them would have greater mildew severity, probably due to more spore arrival, or just conditions are better for mildew. We expected that there would be an urban heat island effect detected. The sites with greater urban area would be warmer, that heat would be bad for mildew, that shade would be decreasing temperature, and that shade would also be indirectly promoting mildew through a UV filtering effect, and lots else. Okay, so that's our hypothesis. Here's our results. So here I'm showing the % urban area in the 1,000 meters around the garden. One big thing was that the more urban gardens had bigger leaves, bigger plants, more leaves per plant. which bigger plants also hosted greater mildew severity. Temperature measured across the full 24-hour period was bad for mildew severity, which is, I think that's what we hypothesized, right? Yeah. Shade, decreased temperature. It's always good when physics works in an experiment like this. And a few other things. But to make our lives more complicated, we also measured that percent urban area at 2 different scales, because we really weren't sure, like, is it the kind of 1,000 meters around a garden that matters for that urbanization context, or is it the super fine scale, like the 50-meter radius around? And what you can see from those bar graphs on the left is that, so the bottom bar graph is the 1,000-meter scale. And if we shrink our zone of urban metric to only 50 meters, we see a little flip-flopping of, one of the most urban site at the 1,000-meter level actually has quite a bit of green space right near the garden, which brings its small-scale urbanization context down. And also one of our rural sites, which is in a very forested and prairie context, actually has a big amount of impervious surface pretty close to it, which is why its urbanization metric goes up. So now, brace yourselves, I'm going to show you what we're kind of currently grappling with in these results, is that we have 4 different structural equation models with very qualitatively different outcomes depending on whether we compute our urbanization metric at the 50-meter radius, the top 2 models, or the 1,000-meter radius, and also whether we focus on the temperatures measured at nighttime only, the right 2 models, or full 24 hours. And so we're still kind of figuring out how to present these in a paper, but it is really interesting that we see, for example, in the bottom right, I'm just going to highlight like 2 things and then move on to the end of the talk. But the bottom right, at the 1,000-meter scale, we do see an urban heat island effect at night. At the 1,000-meter scale, we do not see an urban heat island effect at that 24-hour period. So that's reassuring. It's consistent with what the timeline when people see urban heat island effects. Interestingly, we also see that urbanization only directly promotes mildew severity when we quantify it at that very fine scale in the top left graph. I said we also tested, so we'll kind of return to this. So we also tested whether the urban plants are simply more susceptible to disease. Well, actually, we tested whether they're whether the pathogens seem to be locally adapted, which they're not. There's no significant plant line origin by garden context interaction. But there was a little bit higher infection severity on the urban plant lines. Not like super higher, but they got a little bit more infected. I will say we've also done other infection assays in the lab to show that there's not like qualitative resistance differences between this, urban, suburban, and rural plants. They all can get infected by the mildew strains that are occurring in our area. It's just in these gardens, some got a little more infected than others. So where we're at with these conclusions are that there are both direct and indirect effects of temperature and urbanization on mildew severity. Some of those effects were like consistent, no matter how we sliced and diced the data in our model structure. But the paths involving urbanization and temperature were really sensitive to the spatial scale of the urbanization metric and the time window of the temperature average. So we have some more thinking to do of how to explain that. The plants from urban seed origins had somewhat greater mildew severity, regardless of what garden we placed them in. Okay, so to just summarize, we have some super cool powdery mildews that infect Plantagos with some species specificity going on. Powdery mildew does occur on Plantago all over the world. At least in our part of the world, we're detecting a signature of local adaptation to temperature. And we have some interesting things we can do with these 2 species that differ in their mating system between the homothallic and heterothallic fungus. Shade is really important to powdery mildew in St. Louis. We are eager to collaborate with folks in other cities to test whether shade matters more in some places than others. For example, in a not so sweltering place as St. Louis, is shade less of a strong promoter of mildew severity. But hopefully, I made you fall in love with Plantago and powdery mildew, or at least convinced you that it's worth studying wild plant pathogen systems, in part because you can test things about climate variation at different scales. Some fungal pathogens seem to harbor substantial regional variation in thermal performance that may aid their adaptation to warmer climates. And people should study plant diseases in urban contexts, because urban agriculture is important. Plants are really important to humans for a variety of reasons, and we don't know nearly enough about when they get infected and how much. And microclimates are important. Okay, so to wrap up, I want to acknowledge the wonderful members of my lab who contributed directly to these projects, as well as our key collaborators on these projects, our funding sources, the lots of undergrads and high school students who have worked with us over the last several years on these and other projects, and then importantly, the folks who let us walk around and stare at the ground and collect data on plants and set up gardens at their schools, community gardens, and research stations. So thanks so much, and I'd be happy to take questions if there's time.
Who wants the mic? It is working, it's just quiet.
Thank you so much. This is such a cool study. I was just wondering, have you considered just like the factor of humidity or rainfall or water splashes in the urbanization experiment? Because powdery mildew is like, they need high humidity, but they are very scared of water splashes. Could it be just like the shaded place, just the leaves won't get splashed with water, and the sun,
yeah, we have definitely thought about that, but we don't have a good way. Well, we haven't measured it yet. Then I guess we could just sit and collect water and see how much. Yeah, I don't know if we would get it kind of quite the force of water impact. Probably there's a way to do it.
I feel like kindergartners, right? Like have them splash stuff.
Yeah. Oh, yeah, we could do that. In our current experiment, I don't know if we could.
Do this with the preschool. Like. Yeah.
The schools could definitely do it. No, it is a very good point. So our data loggers measure temperature and soil moisture. So we have some moisture data to work with. But you're right, powdery mildews need sort of like moderately high humidity, but not direct water, at least not when they're germinating. It can impede their germination. In our time series, the precipitation and humidity don't seem to be important for explaining year-to-year variation in epidemic size, but it's one of those things. It's hard to figure out exactly what time window, what time lag to focus on. Is there a critical month when humidity or precipitation matters? But yeah, experimentally, yeah, there's a lot we could still do with splashing water on plants at different times, for example.
Urbanization is real, and then there are differences between treatments. I think that's cool.
Yeah.
My question is about fungal mating systems and whether or not the plant hosts that they're kind of specialized on have anything interesting with their mating systems, or genetic would explain the pattern.
Yeah, so interestingly, so the question is whether the plant mating systems have anything interesting that kind of tracks with the interesting mildew mating system. And yes, but in a weird way. So Plantago lanceolata is self-incompatible. Its mildew is self-compatible. Plantago regelii and major are both, as plants, self-compatible, but their mildew is self-incompatible. So I don't really know what to do with that, but it is fun to think about from a kind of population genetics perspective. Yeah, so I guess they have kind of opposing tendencies towards novel genetic recombination through sex, at least.
One last question.
Got pressure. I was thinking more about the urbanization. So you mentioned that the more rural areas are more shaded because of the trees. When I think about cities, it's like you get a lot of buildings and then a lot of shade. And I think Plantago often grows like wherever, like next to the concrete pavements and like by the edge of the buildings. And so I'm wondering, do you think like in the micro scale in urban areas, could there be like hot spots for spores that are spreading?
I absolutely think that. Actually, that I probably should have clarified that in my point. I think that's exactly what's happening. Is I think that the microhabitats are really critical to the dynamics in this system. That there's probably just some really favorable, persistent sites of infection. Like it is often like along the edge of a building where nobody. I think mowing is a big part of it too. Even though we showed that mowing isn't everything in explaining the shade pattern, shade definitely matters on its own. I think mowing contributes. We also have some mowing data to back that up. That mowing and mowing could contribute in different interesting ways, because mowing, of course, is like a culling of infected or uninfected host tissue. It may also help disperse spores a little. And I have some fun ideas for experiments we could do to test that, and different types of mowers, and whether, like, these are a lot of parks that we study. Is it contracting with like particular mowers that then travel from park to park with their equipment? Are they helping bring spores from park to park? Maybe, but definitely I think the mowing is really important to the mildew persistence, and Plantago, I think it could be one reason why we don't see as much disease on Plantago lanceolata is their leaves are sort of more easily prone to being hacked off by a mower. Regalia tends to be a bit flatter to the ground, and we'll see mowing damage on the tips of the leaves, but a lot of the leaf is a bit flatter to the ground and escapes. From the plant side, there's also the lanceolata seed stalks. The stems are much taller, and then the seeds are all at the tippy top. So we often never find mature seeds of lanceolata in mowed parks, whereas Regalia's seeds start closer to the ground, and a lot of them survive being decapitated by the mower. So I think mowing is a really interesting thing in general.
I think we'll end with plant decapitation.
Plant decapitation.
Thank you, Rachel.
Thank you. It's a good way to decapitation.
You can't top plant decapitation.
Is this?